a DB Hume Early Career Fellow at the University of East Anglia in the UK. And I'll be talking today, giving you an overview of my work exploring determinants of reproductive success and failure. So full disclosure, I'm a behavioural ecologist who just happens to have worked on several hemipteran systems whilst trying to address questions that get at determinants of reproductive success. And when I'm talking about reproductive success, I'm primarily focusing on how organisms go about producing offspring. And there's several components that are a part of this pathway to reproductive success. Firstly, there's the pre-copulatory element. So before mating occurs, individuals are under selection to find mates. And that's resulted in these really elaborate traits that have evolved, such as the weapons that we see of deer, these elaborate tails and ornaments that we see on widow birds and on peacock spiders. But the pathway to reproductive success doesn't stop at the point of mating. There's also a lot of elaborate morphologies and behaviors that have evolved in order to maximize the chances of fertilization success and the chances of siring offspring. And that sounds fairly straightforward, but during my PhD, I came across really high levels of mating failure. And this seems counterintuitive because individuals are under such strong selection to produce offspring. You'd assume that instances of infertility, potential sperm depletion, or genetic incompatibility might be very low. And in fact, mating failure is surprisingly common. This is a panel just taken from a short review. And in the review, it was noted that a species that has particularly high levels of infertile matings, upwards of 50% of matings that don't produce any offspring, is the seed bug, Ligeus simulans. And this was the topic that I went on to explore over the course of my PhD. And looking across 5 different seed bug species, I found really surprisingly high levels of mating failure. So in green are the percentages of pairings that actually produced offspring. You can see that for 4 species, that's around 50% of matings. And in the instance of Ligeus creticus, there are actually less than 15% of the matings that actually produced offspring. So we were seeing really high rates of mating failure. And taking a closer look at this, I was interested in whether specific individuals were perhaps worse at producing offspring than others. And so I looked to see if this mating failure was repeatable. And to do that, I gave the same males multiple chances to produce offspring with 4 different females, and found that overall, again, around half of matings didn't produce offspring, but also that 26% of the males in this experiment produced no offspring despite being given 4 separate chances to do so. So this appears to be a male-associated and repeatable phenomenon. And a potential clue came when we were able to do some micro-CT imaging of these insects whilst they were in copula. And you can see here, this is a male at the top of the image and a female at the bottom, zoomed in on their genitalia. At the top, there are the male's claspers, in blue, the parameres, and then the oedagus with this really elaborate elongated processus in pink. And that is then inside the female reproductive tract with the female sperm storage organ, the spermatheca, in yellow at the bottom. And this complex elongated genitalia means that it actually takes a very long time for males to be able to get to the point at which they can successfully transfer sperm, meaning that if matings are too short, then sperm transfer doesn't occur, and we see this infertile phenotype. And these are just some images across several of the species. At the top, we have the male processus, and in each of these instances, they're very elongated. And then at the bottom, we have the corresponding female anatomy. With the arrow, that's showing the valve that regulates the entry and exit of sperm into the female sperm storage organ. And the females have fairly convoluted sperm storage organs as well. You can see this sort of coiling morphology here. And so it takes upwards of 30 minutes of mating before the processus reaches the valve in the female reproductive tract and sperm can be transferred. Another really surprising side effect of having such a elongated genitalia that I found when I was doing dissections, you can see from this striking image here. And this is the female reproductive anatomy, and you can see with this F here, this denotes a fragment of a male genital processus that has actually broken off during mating and been left inside the female reproductive tract. And from a study from colleagues, 25% of males that have had mating experience actually have some evidence of this processus or flagellum breakage that is visible. So mating in this system seems to be quite a protracted and complex process, and that's resulting in these quite high rates of mating failure and low fertility, which is still somewhat of a puzzle to try and disentangle. But I've continued to be interested in widespread variation in reproductive success and asking why more individuals aren't more reproductively successful given the selection on them to maximize their offspring production. And more recently, during the course of my last postdoc, I was working with these charismatic Mitrans, the Coreyids, leaf-footed bugs, and specifically for the majority of the time with Narnia femerata. You can see two males here that are fighting. They feed on this cactus fruit, and males have these spiny hind limbs that they engage in this contest behavior with, where they grapple with each other to monopolize access to cactus fruits and the females that come and visit to feed on that fruit to then subsequently mate with them. You can see from the images along the top that there's a really wide variation in levels of exaggeration of this hind leg weapon. Some species have these large tibial spines. This Acanthocephala has this massive hind tibia here. And then other species, such as the Anassa squash bugs, have very reduced, hardly any exaggeration whatsoever. So a lot of variation to work with across species. And when I was looking at rates of infertility and hatching failure in Narnia femerata, we don't see any of the same severity of reproductive failure in the species. So the majority of eggs all hatched up here. So this is 100% hatching rate. Most individuals were producing clutches that all hatched. Interestingly, a couple of females produced clutches where no eggs hatched, and there were some individuals that had incomplete hatching success, kind of in the middle. So we're not seeing complete hatching success in this species either. So nowhere near as extreme reproductive failure, but still non-trivial rates of hatching failure. And alongside this variation in hatching success, we found really high levels of variation in testes mass just within the species. So this plot is showing the allometric scaling between body mass of males and their testes mass. And we see a 17-fold variance in testes mass in the species. And so if larger testes can produce more sperm and can maximize the chances of fertilization, why don't males invest in larger testes and therefore guarantee the chances that they're going to fertilize a female's eggs? So the answer to this is potentially somewhere in this figure, which is showing the metabolic rates, or so the oxygen consumption of different metabolic products in the muscular hind legs in gray and also in the testes in orange. And basically, all you need to take away from this is that testes tissue has very similar, if not higher, levels of metabolic activity than this costly leg muscle tissue. So testes tissue is potentially equally, if not more so, expensive to invest in and maintain. And this can lead to a potential trade-off. As I mentioned, individuals can invest in weapons, so their hind legs in these careers, and also testes to maximize the chances of offspring production. But if both of these things are expensive, then investing in weapons might come at the cost of testes and vice versa. So we have this kind of pull between these two different categories of traits. And we can envisage this as a trade-off that's preventing individuals from maximizing their reproductive success in this system. And there's some notable previous literature demonstrating the presence of trade-offs between pre-copulatory and post-copulatory traits. Most strikingly in dung beetles, where ablation of the weapon, the horns, results in increased testes size, suggesting that investment in the two is linked and one comes at the expense of the other. Up to now, most of this research has been quite correlational. And so what we wanted to do was make use of a really striking behavior that Koreans can partake in, which is autotomy. And we can use that autotomy, which is voluntary limb loss, to see if we cut off the investment in their weapon, their limb, does that influence their investment in post-copulatory traits for their testes? And so, yeah, we can experimentally try and get at this potential trade-off between weapons and testes by inducing weapon loss. You can see in this video here, just by gently gripping the hind leg, the weapon, the insect voluntarily releases the hind leg at a point at an inbuilt plane of fracture and walks away. So yeah, we can use this autotomy behavior to manipulate the trade-off between weapons and testes. And the capacity to autotomize is very widespread across Koreids. So from this phylogeny of Koreids, the colors just denote the speed of autotomy, fast being red and slow being greens into blues. But pretty much all of the species can autotomize at some speed or other. And we already know that autotomizing individuals when they're juveniles does give them a boost in testes size. So if we remove one of their weapons when they're still developing, as adults, they will have larger testes. And I wanted to see how flexible this morphological trade-off was by inducing autotomy earlier on in development, in the 4th instar and the 5th instar, and then also during adulthood to see if individuals could continue to balance investment in weapons and testes into adulthood. And quite clearly, we see that if you autotomize individuals as juveniles, then you do see this increase in testes mass, but this doesn't occur past the point of adulthood. So adults that lose a limb don't have the same boost in relative testes mass that we see if this happens during juvenilehood. And interestingly, because we can control the loss of the limb via autotomy, we can kind of directly see how the mass of the limb that we prevent the insect from growing translates into a potential increase in testes size. So when we autotomized the individual, we weighed the limb, and then they went on to grow a full-size limb on their other hind weapon. And we can subtract the mass of the limb we removed from the one they went on to grow and use that as a proxy for the amount of mass that we sort of stopped them from growing through the process of autotomy. So you can see when we autotomized them as small juveniles, 4th instar juveniles, there's a really big difference between the limb that we removed and the one they went on to grow. And when we autotomize them as fully grown adults, there's no significant difference because that's a fully grown limb that we've induced them to drop. And the bigger the difference in mass between the final limb they grew and the one we removed, the bigger the boost in testes. So we see this striking positive correlation here. And this correlation is positive at the 4th instar and the 5th instar, but we see no differences during adulthood. Again, just emphasizing that this trade-off seems to be mediated exclusively when they're juveniles, and we don't see any differences in allocation past that point. So just to summarize that, eliminating investment in weapons in Narnia Femerata does boost testes mass, but only if it occurs during the juvenile stage. So this autotomy is evidence for direct reallocation. We're not just kind of triggering them to reinvest in their testes if they've lost a weapon. It's actually seemingly a reallocation of resources from the limb to the testes. And males crucially have to balance this investment in their pre-copulatory traits, their weapons, with their post-copulatory traits, their testes. And then finally, I just want to show like a quick overview of how this plays out in terms of mating system dynamics from some experiments I did in semi-natural enclosures. And here you can see one male Narnia Femerata with 4 females. This is the cactus fruit that is the resource that they feed on and that the males defend. And I observed males over a 5-day period and females as well in these enclosures. So I have 16 replica enclosures with 8 males and 8 females in each. And using an automated imaging camera system, I could track them over the course of this time period and record any mating events and their locations within the arena to see actually what the kind of behavioral dynamics are of these individuals under these natural conditions. And just looking at the skew of mating success, the number of mating partners males have, just ignoring the red and the blue different treatments, we can see that the majority of males just were never seen mating over the course of 5 days. And only a couple of males had multiple partners, so we're seen mating with 3 or more females over the course of the 5-day experiment. So there's this really strong skew, as we would expect from a resource defense mating system, where males are using their weapons to monopolize access to the resource and preventing other males from mating. And again, we also see a skew in female mating rates. So the majority of females, again, didn't mate or weren't observed mating during this experiment. And only a couple were seen mating with more than 1 male over the course of the experiment. And I mentioned this diversity in weapons, the spectrum of weaponization across careers. And so to emphasize the differences that we see across this taxon, whilst we have resource defense mating systems and really skewed mating success distributions in Narnia Femerata, doing a similar experiment in squash bugs, specifically in Anassa tristis, we see quite different patterns. So they have no obvious weapons, and therefore we wouldn't expect any sort of resource defense behavior. But how does that play out in terms of their mating success distribution? So under very similar conditions, tracking them for a slightly longer period with 10 males and 10 females in each enclosure, every individual was numbered and we could track their mating interactions. And we see something quite different. We see that we have a sort of normal distribution of mating success in both males and females. Lots of mating activity in both sexes, and the majority of both males and females mated at least with half the individuals in the arena. And when we visualize this as a network, this is just one example with the males, 10 males in the arena along the top and the 10 females in the arena along the bottom. And we draw lines between every individual that we saw mating. We can see this really kind of complex mating network that built up over the 10-day period. And there's a huge amount of mating activity, and pretty much most individuals are mating with pretty much all the other available mating combinations in the arena. So yeah, in this case, up to 68% of all available combinations between the individuals were realized over the course of the experiment. So that's very strikingly different potentially predictable because they don't have this obvious weapon, and so our assumption that they wouldn't have any resource defense behavior was met in this case. They've got much lower variance in mating success and really highly polyandrous mating system dynamics. And so that was just a quick overview of the widespread variation in reproductive success and how hemipteran systems have helped shed light on this in my research. From one end of the reproductive success spectrum, looking at causes of mating failure, low fertility, and reasons potentially why offspring production isn't happening, through to the other end, trying to work out how individuals are maximizing reproductive success, what trade-offs are preventing them from doing so, and what kind of mating system dynamics are at play across different species. And I'd like to acknowledge my postdoc and PhD supervisors and lab groups, and a lot of undergrads who were super helpful with data collection. And hopefully there's time for questions. Thanks.